Hello students, today's topic is animal cells as a replacement for the whole animal in toxicity testing. Toxicology is a branch of science that deals with toxic substances and poisons. Toxicity testing is an essential step in screening of new drugs in drug designing and development programs to evaluate toxic effects of chemicals or substances on human, animals, and environment. A toxicity test is designed to generate data to check the adverse effects of substances on human or animal health or environment. Certain endpoints are designed for each test. Traditionally, toxicity test has been based on studies on experimental animals, especially rodents. The tests are based on test duration, life stage, and endpoints. Interpretations are made based on responses of organisms to different concentration of test compounds and control. They are categorized as acute, subacute, or chronic tests based on the duration of exposure of the test substance. Acute, subacute, and chronic test studies are done to estimate toxicity endpoints. Example, LC50 value or IC50 values. We have discussed in detail about these terms and tests earlier. In recent years, there has been an increasing concern over the use of animals in toxicology studies. The 3R strategy was proposed with the intention of using alternative methods for toxicity studies without compromising with scientific results. The 3R stands for refinement, reduction, and replacement. Refinement to redefine experimental procedures which would decrease unnecessary pain and trauma to the animals. Reduction to reduce the number of animals for the experiment, and replacement to replace animal experiments with computer models, cell line, and in vitro methods. Cell line systems have been an alternative to animal study in toxicology tests. Both primary and cell lines can be used in toxicology study. The source of cells could be avian, rodent, or human cells. Certain 3D cell models like spheroids have also been proved to be good models for toxicity studies. Example, MCF10A cells or the human liver cell line. Certain cell lines used in general toxicity studies are diploid human fibroblast cells, HeLa cells, or tumor cell lines. The general morphological changes observed in these cells when exposed to toxicity are noted. Example, blebbing, vacuolization, fine ultrastructural modifications. Certain other parameters noted are cell viability, cell growth, metabolic processes like O2 consumption or NADH, NAD conversions, etc. Some of the cell viability studies like Triton Blue and Evans Blue are discussed earlier. Thus, the effect of toxic substances on cells can be estimated by evaluating the basal functions of the cell like vital staining, cytosolic enzyme release, cell growth, etc. In certain cases, using certain specialized cells for specialized cell functions. Example, beating heart rate in myocytes or myocardial cells. Assays to test cell death are generally used in toxicity tests. It involves treating cultured cells with varying increasing doses or concentration of test substances and use one of the many methods to quantify the live or dead cells. It is also used in making dosage curves in pharmacology. The endpoints at some percentage of cell death, example, the most commonly used LC50 or IC50, is then used to compare the relative effect of different test compounds. The common assay used are lactate dehydrogenase leakage assay, reduction of tetrazolium di-MTT, neutral red uptake, tritium labeled thymidine uptake, and fluorescent assays to check fluorescent permeability across cell layers. We'll be discussing the first four assay methods in detail. Lactate dehydrogenase assay. Lactate dehydrogenase is an intracellular enzyme which catalyzes the reversible conversion of pyruvate to lactate via reduction of NAD to NADH. It is a final step in glycolysis. It is found in high levels in all cells or tissues that use glucose for energy. Increase in LDH level in cells indicate damage. When cell damage occurs, this enzyme is released into the blood. In case of in vitro testing methods, it is released into the media. The released LDH is then quantified by certain enzymatic reactions. One of the method is by use of tetrazonium salts. In this case, the cell lines when treated with the test substance, if damaged, release LDH into the media. LDH catalyzes the conversion of lactate to pyruvate by reducing NAD to NADH. The NADPH dehydrogenase diphorase uses NADH to reduce a tetrazonium salt INT to a colored component. This colored component has absorbance and quantified at 490 to 520 nanometer. The absorbance is proportional to the amount of LDH in the media, which can then be correlated to cell damage by the cell substance at that concentration. The figure here shows a schematic representation of a lactate dehydrogenase assay. MTT assay. It is a calorimetric method to measure the reduction of yellow-colored tetrazolium salt to a purple-colored formazan by mitochondrial succinate dehydrogenase enzyme. MTT is a water-soluble component and penetrates into cells, while formazan is insoluble and remains entrapped in cells. This is made soluble by means of DMSO or isopropanol and then quantified at 570 nanometer. Dead cells lack active mitochondrial dehydrogenase. Hence, the conversion of tetrazolium to formazan does not occur. Hence, MTT assay is used to determine viable cells. The figure here is a schematic representation of an MTT assay using a kit. Calcine assay. This assay helps to check cell viability. The dye, a acetomethoxy derivative, can be transported through the cell membrane of live cells. In the cells, the intracellular esterase enzyme removes the acetomethoxy group and the molecule gets entrapped inside. It gives a strong green fluorescence which can be quantified by flow cytometry. Dead cells lack active esterases. The figure here summarizes the three assays that we studied. LDH assay, MTT assay, and calcine assay. Neutral red assay. It is based on lysosomal activity. The neutral red dye is a weak cationic dye which can easily penetrate into the cells by non-ionic diffusion. It gets accumulated in the lysosome. Here, it binds to the anionic sites of the lysosome matrix. Hence, it determines the membrane permeability of the cells. The major drawback of neutral red assay is that it cannot distinguish between cells which are already dead and in the process of dying. Tritium labeled thymidine uptake assay. It measures the rate of proliferation or cell division to check the effect of the test substance. The cells are cultured in wells followed by addition of the test substance. Thymidine is added to the cells. The thymidine uptake by the cells is proportional to DNA replication. The amount of tritium in the nucleus is then quantified. It is a sensitive method, however, the radioactivity is a concern. Hence, an alternative to this is the bromodeoxyuridine assay. The principle is same. The incorporation of bromodeoxyuridine is assayed by means of antibody. The schematic representation of tritium labeled thymidine uptake and bromodeoxyuridine assay is given here. Cytotoxicity tests using specialized cells have proved to be useful if the in vivo toxicity test with the chemical is already established, and in vitro investigations using specialized cell cultures has also been done to study the toxic action of the test material on the target tissue. These tests provide an insight on the pathogenesis of some human diseases like liver diseases.